Hello everyone and welcome to video 10. Well done for getting this far. So we should be experts on enzymes, so we've got our first starter activity. But before we get on to that, we're going to move on to key area 5, which is genetic engineering. So if you want to print out the activities and consumables and see the PowerPoint, you need to go into 1 cell biology, and then you want to go to 5 genetic engineering. Then you're going to do the starter activity, so you might want to pause the video and try to answer the questions, and then I will reveal the answers to you. So, the question says the diagram below shows the action of enzyme phosphorylase in a potato cell, and then you've got glucose 1-phosphate with phosphorylase going to starch. It says underline the option in the brackets to make the sentence correct. The action of the enzyme phosphorylase catalyzes and it's either synthesis or degradation of starch. For II, you've got state the effect of phosphorylase on the rate of this reaction, and then for free, so you've got explain why lipase could not produce starch in this reaction. So the answers to that are that it's a synthesis reaction because it's the one synthesis reaction that you need to know. And the effect of phosphorylase on the rate of reaction, well, phosphorylase is an enzyme, so it speeds up the rate of the reaction. And then explain why lipase could not produce starch in this reaction. So we know that enzymes are specific. So lipase is specific to one substrate, which is not starch. So its active site is complementary shape to its own substrate, so would not be able to fit the starch. So today we're going to learn about genetic engineering. So your learning intentions are to describe genetic engineering. You need to be able to state the stages of genetic engineering. You need to know what some of the products of genetic engineering are, and you need to know some of the controversy surrounding genetic engineering. So to be successful, you'll be able to explain what genetic engineering is. You'll be able to describe each stage of genetic engineering. You'll be able to label a genetic engineering diagram, and you should know some of the products of genetic engineering. So genetic engineering is the process of transferring genetic information from one organism to another. So the organism which receives the genetic information is described as a genetically modified organism, or a GM organism. You'll need to make a note on this, and you need to know it. So you'd put the head in genetic engineering, and then you'd put the bit below. So you could write it out many times, or you could try to speak it many times. So bacterial cells are often used in genetic engineering, and that's because you can multiply them rapidly, and also they're cheap. So they are different from your animal, plant and fungi cell. So what I want you to do is I want you to write down the differences so that you can remember about your bacterial cell, and pause the video while you're doing this before I go over the answers. So you should be able to remember this from video 1. So bacteria cells do not have a nucleus, so instead they have to have something else. So they have their circular DNA, and they also have these things called plasmids, which contain DNA. So the plasmids can be removed from the bacterial cell, and it can be used in genetic engineering. So your plasmids just now are super important for what we're about to talk about. So your polo shapes. So you've got different stages in genetic engineering. So our animal cell is different to our bacterial cell. So in our animal cell, we have our cell membrane, which is in our green, and we have our cytoplasm, which is our blue. And then we've got our circle, which is our nucleus. And then we've got our yellow things, which are our chromosomes. And then we've got a red part, which would be our gene, which is your section of your chromosome in the section of DNA, which encodes a protein. So for the first stage, we need to identify the gene that we need. So for example, this might be an insulin gene. And then we need to identify where this gene is located, so what chromosome it's located on. And then we need to extract the chromosome where we have the gene. So the chromosome's removed, and then we've got to cut the desired gene out of the chromosome. So we use enzymes to do that. So we cut the gene, and then we're left with just the gene that encodes for the protein. So for example, insulin. The next stage of genetic engineering is that we extract a plasmid from a bacterial cell. Then what we do is we cut open the plasmid and then we insert the gene with different enzymes. So the plasmid is cut open using enzymes, and then the required gene is inserted into the plasmid, again using enzymes, but different enzymes. And then once we've got this desired new plasmid, which has got our new gene inserted, then the plasmid with the desired gene is then inserted into a different bacterial cell. So your bacterial cell with your plasmid in it. So the bacterium now contains a plasmid which contains a gene which was not in the original bacterium, so making it a genetically modified organism. So in fact, it is usually from a completely different species. So in this case, our gene was inserted from an animal cell. So we can imagine, like, for example, the insulin gene. The bacterium can now be put into a solution which provides all of its nutrients it requires. So for example, nutrient agar or nutrient broth. So if we put the bacterial cell onto our agar plate and our nutrient, so the bacterium can now reproduce, and each new cell will contain a copy of the new gene. So bacteria can only reproduce very quickly in ideal conditions. So after a very short time, there will be many bacteria, each capable of manufacturing the protein code for the gene. So then you would want to then purify your gene so that you didn't have any bacterial cells with it. So you would then purify your gene and then you would make your product. So this is just a summary of genetic engineering of what we have said. So first of all, you have your animal cell and you need to identify your desired gene. So you have to locate it on a chromosome. So then you extract the chromosome and then you cut out the gene. So we've got our human insulin gene here. Then you get a bacterial cell and you extract a plasmid. And then you cut your plasmid open with enzymes and then you insert your desired gene, so your insulin gene, into the plasmid. And then you put your new plasmid that you've just created into another bacterial cell. Your bacterial cell is then grown in nutrient conditions. So for example, on a nutrient agar plate. And then after you've done that, you've got your bacteria multiplies in a, like for example, a fermentation tank. And then you would then extract and purify your human insulin. And then you would be able to make masses of your product, human insulin, and help people that had diabetes. So if you want to, you can watch this YouTube video which further explains it. You won't be able to click the link, but you'll be able to pause the video and go to the link from the other PowerPoint. So this just summarises what we've said. So genetic engineering is when genes from one organism are inserted into another organism. So e.g. genes from a human are inserted into a bacterial cell. So you have two videos here that you can watch. So you've got one on insulin and one on genetic modification. So for your first stage of genetic engineering, you have to identify your desired gene. So for example, human insulin. For your second stage, you need to extract your gene from the chromosome. And for the third stage, you need to get your plasmid from your bacterial cell. And then you need to cut it open and then insert the gene into the bacterial plasmid. Then you put your plasmid with your desired gene into another bacterial cell, which can be called a host cell. And then you want to make the bacterial cell grow and produce lots of your desired gene product, which would be insulin in this case. So for bacteria to grow rapidly, you need to put them in nutrients. And then this will mean that you have your bacteria with this desired product and plasmid with the desired gene growing. So organisms which have had an additional gene inserted into them are called genetically modified GM organisms. And the stage that's missing here would be that you would have to purify your product. So again, summarising this, but this time in diagram form. So this would be your first stage where you identify your desired gene. So for example, insulin on your chromosome, you locate your chromosome with your desired gene, you then extract your chromosome, you then cut your chromosome and you get your desired gene, and you cut your chromosome by using enzymes. And then you'd get a plasmid from a bacterial cell. You'd then cut open the plasmid using enzymes, and then you would insert your desired gene into the plasmid by using enzymes, and then you'd put the plasmid containing the desired gene into another bacteria, and then this bacteria you'd put into nutrients and ideal conditions, so then it would grow rapidly, producing lots and lots of your desired gene with your plasmid. And then you would have to then produce insulin, but before you could do that, you'd have to make sure you extracted it and purified it. So you need to try, from what we have just said, to put the following steps in the correct order. So it's best to pause the video and try to write these stages in the correct order before I reveal the answers. So the answers to the stages in genetic engineering are: you'd identify the section of DNA that contained the required gene. For stage 2, you'd extract the required gene from chromosomes using enzymes. And then for 3, you'd extract the plasmid from the bacterial cell. For 4, you'd then cut open the plasmid and insert the required gene into the bacterial plasmid using enzymes. Then for 5, you'd insert the plasmid into the host cell. And then for 6, you would grow and multiply the GM bacteria in nutrients. And then for 7, you'd get your desired product produced by extracting and purifying it. So they're the stages of genetic engineering, so you need to write them down many times, you need to say them many times, and you need to know them. You can also use Play-Doh to model it, or you can use, you could draw pictures of it, but you need to know all of the stages of genetic engineering, because quite often you have to put the stages in order, or you have to label diagrams. This could be a question, so you need to label the different parts of this diagram. So this is going to be what type of cell. So well done if you got animal cell. And what is this part here going to be? So it's been extracted, so that's going to be your chromosome. And then what part is taken out of your chromosome? It would be your desired gene. For this cell here, it's going to be your bacterial cell, so well done if you got that correct. And the polo shaped thing down here is going to be your plasmid, so again, well done if you got that correct. Then for this one, you would have to do what to your plasmid? You'd have to cut it open with enzymes. And then you would have to insert your identified gene, so your desired gene, into your plasmid, and you put it into a new bacterial cell. So your bacterial cell would contain your plasmid with your desired gene, and then you'd want it to grow and multiply, and then you'd extract and purify your product. So what is used to cut and insert the gene? We should know that the answer to that is enzymes. Another common question that you might get asked is what protein is produced by genetic engineering, and you need to know that two things are commonly produced by genetic engineering. So these two things are human growth hormone and insulin. So you need to write that down. So in your activities and consumables, you should find a cut and paste exercise that you should be able to complete now. So we've looked in detail at genetic engineering involving bacterial cells, but genetic engineering can also involve multicellular organisms. So in recent years, scientists have managed to inject a plasmid containing a required gene into the cells of multicellular organisms. So examples of multicellular genetically modified organisms include golden rice, blight-resistant potatoes, and tomatoes with a longer shelf life. So I'm going to take you through each of these examples, but you do not need to write anything until we get to the table. So don't write all of the information down. This is more detailed than you need to know it in. So let's start off with golden rice. So vitamin A is a chemical needed in small quantities in the diet for good health and in particular for good eyesight. So normal cultivated rice contains very little vitamin A. So in parts of the world where rice is the main part of the diet, vitamin A deficiency is common, and this can lead to vision problems and even blindness. So genetic engineers have transferred two genes from other plants into rice to create a variety called golden rice, and it gets its name because it's a golden colour. This new strain makes a yellow chemical which gives them a golden appearance, and when the rice is eaten, the yellow chemical is changed to vitamin A in the body. It also contains a higher percentage of iron, which is needed for the production of new red blood cells. So this will help to stop people in developing countries and prevent them from becoming blind or visually impaired. Another multicellular organism which has got its genes altered would be the blight-resistant potatoes. So potato blight is caused by a parasitic fungi that ruins potatoes and makes them inedible. This caused the Irish potato famine in 1845 to 1846 and the Scottish Highland potato famine in 1846. So genetic engineers have identified and extracted a gene from a wild potato plant that gave blight resistance and introduced it into other potato varieties to make them resistant, so they no longer were able to be attacked by this parasitic fungi and were no longer inedible. Unfortunately, new strains of the fungus evolved which were able to infect the new varieties of potato. The disease resistance is now known to be more complex, involving a number of interacting genes, and work is now being done to locate these genes and others which affect the quality of the potato and transfer them as a group into other potato plants. The production of resistant GM crops can have significant environmental benefits as little or no pesticides need to be used. And you will go into that into more detail when you come to your Life on Earth unit. So tomatoes with a longer shelf life are the third multicellular organism. So genetic engineers can now insert a gene which switches off the activity of another gene to extend the shelf life of tomatoes. The gene controls an enzyme that breaks down a chemical in the cell wall of tomatoes, making the fruit become soft. This delays the softening of tomatoes without affecting genes that control colour and flavouring, giving it a much longer shelf life before it becomes overripe and spoiled. So these other genetic modifications you don't need to know about, and they're just a little bit of fun. So you've got the glowfish, so zebrafish, which have had genetic information from bioluminescent jellyfish added to their DNA, so they basically glow in the dark. And then you've got the grapple. So genetic cross between an apple and a grape. The grapple was originally designed to provide a much higher vitamin C dose per fruit for third world aid. The majority of the funding for the fruit came from UNICEF. You have poisonous cabbage. So the gene that codes for poison in scorpion's tail has been inserted into cabbage. These genetically modified cabbages produce scorpion poison that kills caterpillars when they bite leaves, but the toxin is modified so it isn't harmful to humans, thus preventing crops being destroyed by caterpillars. So less cow methane. So we know that cows release methane as a result of their digestion, and that causes quite a lot of CO2 emissions, which can lead to global warming. So methane is produced by a bacteria that feeds on the grass and hay in the stomach of the cow. So methane contributes to the greenhouse effect, and scientists have identified the bacteria responsible for producing methane and have produced a strain of cattle that produces 25% less methane than the average cow. So we've got the silk goat, so silk and milk. So strong, flexible spider silk is one of the most valuable materials in nature. It can be used to make lots of products, for example, artificial ligaments and parachute cords. The silk is hard to collect in large quantities, so researchers have inserted a spider's dragline silk gene into the goat's DNA in such a way that the goats would make silk protein in their milk. This is silk milk, and it could then be used to manufacture a web-like material called biosteel. Then you've got large salmon, so genetically modified salmon grow twice as fast as a conventional variety. So the photo shows two salmon the same age, which are genetically altered, one in the rear. So genetically engineered Atlantic salmon has an added growth hormone from a Chinook salmon, which allows the fish to produce growth hormone year-round. Then you've got glow cats. So glow cats are used to help with AIDS research. The cats glow green under UV light because the green fluorescent protein gene originating from the jellyfish has been inserted in them. The GM cats also carry a monkey gene which protects monkeys from the infection by feline immunodeficiency virus of FIV, responsible for cat AIDS. And then you've got an ear mouse. So the vacante mouse was a laboratory mouse that had what looked like a human ear grown on its back. The ear was actually an ear-shaped cartilage structure grown by seeding cow cartilage cells into a biodegradable ear-shaped mold and then implanting it under the skin of the mouse. So there's lots of different genetically modified organisms, and to have a look at more, there's lots. So there's obviously a lot of advantages about genetic engineering because it can produce useful products like insulin, which can be used as medicine. It can produce bigger animals. It can produce animals with abilities to make products that are useful, such as silk. And it can also be used to increase crop yield without using pesticides. And there's lots of things it can be used for. But there's a controversial side to that. So there's lots of things that are against it also. So it's quite an ethical dilemma. So that means that there's things for it and there's things against it. So the things against it might be that people are worried about genetically modifying organisms, because if you genetically modify organisms, then you eat them. Is it going to alter our DNA? Also, are your animals that you've genetically modified, are they going to then mutate and then become not what you want them to be? So there's lots of pros and cons. So if you go into the videos, it'll take you more through that. So there's a table that summarizes what you guys need to know about the products of genetic engineering. So for the treatment of diabetes, we should know that it is insulin. And for the treatment of your dwarfism, so when people are shorter, it would be your human growth hormone. And both of these are made by bacteria. And then for your, it can help prevent blindness by preventing vitamin A deficiencies, it would be your golden rice. And then potatoes are blight resistant, that would be your blight resistant potatoes. And obviously your organisms would be for your golden rice and for your blight resistant potatoes. And then preventing softening and rotting of tomatoes, so giving them a longer shelf life, would be our GM organism, would be our tomatoes, and would be longer shelf tomatoes. So tomatoes with a longer shelf life. So you need to copy this table down and make sure you know them. So there's several advantages to using bacteria and microorganisms instead of multicellular organisms. So the advantages of using microorganisms are that they can be grown quickly, easily, and at low cost. So your GM bacteria can produce large quantities of useful product that can be extracted, concentrated, purified, and put to use. So culturing cells just means growing cells. So in a microbiology laboratory, scientists can work with microorganisms to help cure diseases. So when a certain type of cell is required, so for example bacterial yeast stem cells, cultures may be grown. So they can be grown and then you can look at them under a microscope, and this just shows you a laboratory. So you need to note this. So aseptic techniques would be your title. And this question usually comes up and it says, why do you need to use aseptic techniques? So first of all, aseptic techniques mean just basically sterile and clean techniques. So aseptic, sterile, clean techniques are used to stop contamination and so that it is safe to use microorganisms. So we don't want contamination as it might make microorganisms dangerous. So the precautions and aseptic techniques we use in the lab are because some microorganisms may be harmful. So we have to use these rules and we need to follow them when handling any type of microorganism. And this is to try to create aseptic, sterile conditions so that we are safe. So you need to wash your hands and bench each time you're using them. You need to use sterile equipment. You never open dishes containing microbes, and you dispose of containers and dishes using very high temperatures. So to culture and grow microorganisms such as bacteria, we need to use aseptic conditions. So for example, in a lab, your first step would be to wear a lab coat, so you would be protected and also your clothing would be protected. And then you'd need to wash your hands before and after the experiment to remove microorganisms, so you don't end up having them on your hands and then eating food with microorganisms, or it would make you ill. Then you would use a smooth, non-absorbent work surface cleaned with disinfectant before and after the experiment so that the microbes were killed. You would autoclave or pressure cooker everything basically, so to sterilize all of your glassware. So basically an autoclave heats it to really, really high temperatures above 100 degrees, so it kills all of the bacteria, etc. So that's number 4. Then for number 5, you would use sterile petri dishes of nutrient agar kept closed until they were required to prevent entry of contaminants. Then for 6, if you were going to get bacteria, you would need to flame your wire loop before you use it to destroy unwanted microbes. And then for 7, you would then hold your lid over your petri dish when you are putting the bacteria on the plate, so inoculation, to prevent the entry of contaminants. Then for 8, you would inoculate your dish and you would seal it with tape to prevent entry and exit of your microbes. And then for 9, you'd have your bin lined with your plastic bag with safe disposal of used plates. So it's a special bin that it would go into. And you would not eat and you would not drink in the laboratory. So to make your nutrient agar for your bacteria to grow on, you would need to put your nutrient agar powder and your distilled water into your autoclave. You'd then heat it in your autoclave to 121 degrees Celsius to destroy all microorganisms. And then you would then pour it into your petri dish, but it would obviously need to be a sterile petri dish that you would have had to have autoclaved. So when you're pouring it in, it's important that you flame it and you also hold your lid over your petri dish so that no contaminants can get in. So all equipment must be put in an autoclave and heated under pressure to 121 degrees Celsius. Some microorganisms cannot be killed by boiling water, so that would be 100 degrees Celsius. Autoclaving kills them all. So if you were wanting to transfer your microbe into a plate, for example, this is a fungus that we're using, you would need to flame your wire loop. So you'd need to put it into the Bunsen burner flame and make sure that it went red hot. And then you'd allow your loop to cool. You would then get your tube that was containing your culture of your fungus, or it might be a McCartney bottle, and then you'd have to flame the top of it, so the mouth of the test tube needs to be flamed, which will kill the microbes. And then you would then use the Bunsen burner flame and have your test tube beside that, and then you'd put your loop into your sample. You would use one loop full, and then you would then put your loop onto your plate, and you would continuously hold your lid of your petri dish over it so that it wasn't going to let in any contaminants. And then you would then get your loop and put it into your Bunsen burner flame and let it go red hot again. And then you would repeat. And then you would want to put your adhesive tape onto your plate after you'd finished with putting your sample on. And then you would store them upside down at 30 degrees for 2 days, and you'd let it grow. So the growth of a bacterial culture. So again, you would want to make sure you flamed your loop, and then you would want to flame your test tube at the top to make sure that they were both sterile. And then you would streak your bacteria colonies onto your plate. So then you would then flame your loop again to make it sterile, and you would do the same with the test tube, and then you would plate it. And then once you had enough on your plate, you would tape it down, and then you'd leave it for 2 days. And then you could do the same, but you could put it into a broth instead of putting it onto a plate. So to grow a cell culture, like growing bacteria, you need certain requirements. So your essential requirements are a sterile environment, appropriate growth medium containing glucose and nutrients, a controlled environment to provide optimum levels of oxygen, pH and temperature. So there are lots of videos of genetic engineering and genetic modification that you can have a look at, which talk you through different genetic modifications and the process being controversial, so the pros and the cons. So if you pause the video and have a look through them. And now we've got some past paper questions. So it says, which row in the table identifies the order of stages involved in genetic engineering? So we are wanting to know the first, the second, the third, and the fourth stage. So is the first stage your required gene identified, or is it gene inserted into the plasmid? So if you pause the video and you try to come up with your own answer, so we know that it's going to be your required gene identified. Then for our second stage, is it our gene and plasmid extracted, or is it our gene inserted into plasmid, or is it our required gene identified, or is it modified cells grown? It would be our gene and our plasmid extracted. And then for 3, is it our gene inserted into our plasmid, our gene and plasmid extracted, our modified cells grown, or is it our gene and plasmid extracted? Well, we should know that it is our gene inserted into our plasmid because our plasmid's cut open and our gene's inserted. And then for 4, is it modified cells grown, or is it gene and plasmid extracted, or is it required gene identified? That would be our modified cells grown, so we can produce lots of our product. So the answer would be A. So for the next question, it says genetic engineering can be used to transfer human genes to bacteria artificially. Complete the boxes below to describe each of the steps carried out to transfer a human gene to a bacterial cell. So we've got our chromosome extracted from our human cell and required gene located, and we've got our bacterial cell carrying our plasmid identified. So then we've got step 2. So step 2 for this one, if we've got our chromosome extracted, we're going to have to then extract our gene. And for this next one here, we're going to have to do, because we've got our bacterial cell carrying our plasmid identified, we want to extract our plasmid. Then for step 3, our plasmid would then be cut open and our desired gene would be inserted. And then for stage 4, you'd want to say that your plasmid with your desired gene is inserted into a new bacterial cell, and then your bacterial cell would grow in large numbers. So grow large numbers of bacteria now containing your required human gene. Then we've got name a human hormone which has been produced by genetically engineered bacteria. So that would either be insulin or your human growth hormone. So here is your step 2, which we've said. So your human gene was extracted and your plasmid was removed. And then step 3 would have been your plasmid opened and your human gene inserted in. And then you would have your plasmid that has your desired gene inserted into your bacterial cell. And for B, it would be insulin and your human growth hormone. So the next question says, insulin is used to treat people with diabetes. It can be manufactured by genetically engineered bacteria using the steps described below. So A, bacteria are grown in a fermenter and produce insulin. B, the human insulin gene is isolated. C, the plasmids are removed from bacteria. D, human insulin is purified and used to treat people with diabetes. Then E, the human insulin gene is inserted into the plasmid, and F, altered plasmids are put into bacteria. So it says arrange the step into the correct order by putting the appropriate letter into the empty box. So after B, which is the human insulin gene is isolated, then it would be C, plasmids are removed from the bacteria. So we put C in the box. And then it would have to be E, the human insulin gene is inserted into the plasmid. Then it would have to then be F, so your altered plasmid are put into your bacteria. And then after F, it would then have to be A. So bacteria are grown in a fermenter and produce insulin. And then the last one would be D, which is the human insulin is purified and used to treat people with diabetes. So you should have managed to get all of that right. If you didn't manage to get all of that right, then you'll have to watch video tutorial 10 again. So the answers were B, C, E, F, A, D. So to review your knowledge on genetic engineering and make sure you know everything, you need to go on to Bitesize using the link below and complete the revision and do the test until you get 100%. Then you can complete the cell biology exam practice questions on page 19 and 20, and you can get that through going to the teacher's guide and exam practice. And then you can complete the National 5 textbook testing your knowledge questions on page 45. and then you can complete the How to Pass National 5 Biology questions on page 25 and 6. Then you can complete the Genetic Engineering past paper questions in 4 past papers and mark it until you get 100%. And then the last task, which you really need to do, is complete the Genetic Engineering homework, which is found in free homework. And you need to send it for your teacher for marking so that we can make sure that you're understanding the course properly. So if you finish this video, woohoo, that is us finished Key Area 5. So we're waiting to move on to Key Area 6, Respiration. If you're still struggling with genetic engineering, make sure you watch the video again until you understand everything, because genetic engineering is quite a tough topic. So well done for getting this far.